Hi, I'm Linda Helton.
And I'm Madeline Mariette. We are the co-founders of the nonprofit, the Brain Renewal Project.
Our mission is to raise awareness of brain injury and to raise funds to support cutting-edge scientific research for regenerative medicines to heal the injured brain.
For over a century, it was believed that the injured brain could not heal itself. Unlike skin or other bodily organs can repair themselves, the damaged brain was thought to be incapable of healing. But in recent years, researchers have dramatically changed that idea.
Scientists have discovered that the adult brain can generate new brain cells, a phenomenon once thought impossible. Moreover, cells in the adult brain can form new connections and even help recover lost functions through the brain's remarkable capacity for adaptation and repair.
These breakthroughs have completely changed our understanding of the brain's potential to heal. However, challenges remain.
One challenge is that the brain has built-in barriers for repairing itself. Researchers are now finding ways to get around these barriers. Thanks to new knowledge and technology, it is now possible to find ways to treat and heal the injured brain that were unthinkable just a few decades ago.
And now, we'd like you to meet some of the scientists doing this work.
Hello, I'm David Margulies, co-founder and chairman of Quiver Bioscience. I'm a physician scientist. Over the past 45 years, I've had the privilege of working with great teams on a number of impactful biomedical and bioscientific projects.
Hi, I'm James Fink, Senior Director and Epilepsy Therapeutic Lead at Quiver Bioscience. We work on diseases ranging from neurodevelopment, which include disorders like epilepsy, all the way to neurodegeneration like Lou Gehrig's disease and ALS.
My name is Graham Dempsey. I'm a founder and the chief scientist here at Quiver Bioscience. Our mission is to create transformative therapies for patients with a variety of brain disorders. And we do that by bringing innovative technologies to how we study the brain, and then ultimately how we use that information to inform how we create those medicines.
Hi, I'm Bella Kellis. I am a college student in New York City. Almost 5 years ago, I fell off of a rope swing and hit a rock headfirst, experiencing a traumatic brain injury. At first, it seemed like a very simple injury, nothing more than a torn lip and some bruising on my leg. Shortly after, it became clear that I had much bigger problems and had experienced a traumatic brain injury. It took almost a year to determine the full range of my injury. Some of the testing included EEGs, sleep studies, and blood work. The EEGs were done to determine whether I was having seizures or not. The sleep study was done and confirmed that I had narcolepsy, and then the other studies were done to rule out other problems. Returning to school, I fell asleep in class. I had little emotional regulation, attention problems, and narcolepsy, none of which I experienced before my injury. Over the next 2 years, I received many treatments and therapies which helped remediate the problems I had developed from my brain injury. Since then, I have met with a number of neuroscientists who believe that regenerative medicines will change the outcome for those with moderate to severe brain injuries, and I fully support their efforts. My recovery has been phenomenal, but I have since learned that traumatic brain injury over time permanently impacts 2% of the population, and it disproportionately impacts some groups like veterans.
Brain damage, whether from trauma, stroke, or degenerative disease, remains devastating for individuals and for their families.
Our goal is to actually try and provide what we call disease-modifying therapies for these individuals, or these are medicines that not only manage symptoms, but really transform lives of patients who are suffering from severe diseases.
Now, historically, treatments for brain diseases have been rudimentary, imprecise, or ineffective. We needed a better model.
can see how the cell responded to different dose. That's drug A, and compare that different doses to drug B and see.
That's the other dimension of what we do here at Quiver is we've built technologies that allow us to actually visualize what happens to those neurons.
During the past 10 years, the convergence of genomics, stem cell biology, optical electrophysiology, and advanced AI has allowed us to build human, cell-based models of human brain disease. Quiver Bioscience has now invested a decade and several hundred person-years in scientific and engineering work to build an instrument that can precisely measure the functional activity of human neurons in health and disease. With this instrument, we can measure the effects of disease on human neurons and evaluate the effectiveness of drugs designed to treat these diseases. Now, how does this work? First, we transform an individual's skin or blood cells into neurons. These neurons grow and mature in a dish. They carry a person's genetics, including any genetic abnormality that may cause disease. We then can use these neurons to explore the biology of a wide range of human CNS diseases, including epilepsy, neurodevelopmental disorders, neuropsychiatric disorders, and most recently, neurodegenerative disorders. How do we model a disease using a person's neurons? In essence, we measure changes in the electrical activity of these neurons. As you probably know, neurons' electrical activity is the brain's language. We think, feel, learn, act, and behave because of the electrical activity of our 100 billion neurons and their 100 trillion connections. It's not surprising that brain disease disrupts this activity. It turns out that specific brain disorders cause changes in the electrical activity of these neurons, but all brain disease is not caused by a genetic change, you might say. How does this system accelerate the discovery of new drugs for acquired rather than inherited diseases? Simply, the underlying biology that guides the characteristics of acquired diseases is similar to the biology expressed in inherited brain disorders. So, we create a brain disease model by measuring changes in the electrical activity of neurons that carry disease-causing mutations. We then can design or screen for molecules that can correct or rescue the electrical abnormalities in these diseased neurons. Molecules that correct the abnormalities in the electrical activity of diseased neurons point the way to medicines for the disease.
Why precision therapies? Why drug development? Developing medicines is one of the most exciting and interesting and intellectually challenging endeavors you could possibly imagine. For the brain in particular, it represents an entirely new and exciting level of complexity.
Drug discovery is really long. Sometimes these drugs can take about a decade to make. Here we're spending a lot of time working with cells and patient-derived materials to really screen specific therapeutics for patients in great need. So we go to work every day knowing that what we're doing is really, really hard, but the mission is that important.
It is an incredibly exciting time. We at Quiver are here in beautiful Cambridge, Massachusetts, amongst really an explosion of technologies that are now available to develop different types of medicines. Here what we focused on is a technology called optogenetics. And what does that mean? It means that we can use light to both stimulate the activity of neurons and to record the changes in the activity of those cells. The reason that we do the work we do is because of people and patients, and the whole goal here is to bring technology to bear to create transformative medicines. And one of the things that's so important about the work that we do is ultimately being able to meet and engage the patients, the people who are going to be the recipients of the medicines that we develop. We as a group are so inspired by the patients and the families of the patients that we meet.
It's a really fulfilling job. We get to go work every day and really keep patients at the center of what we do.
Today, the prospect of inducing meaningful brain functional regeneration is becoming realistic. For the first time, we can look for drugs that restore the brain's natural capacity to self-repair. So there's a call to action. We're standing at a turning point in neurology and regenerative medicine. In the next 5 to 7 years, we may be able to see the first FDA-approved drugs capable of reversing brain damage. Imagine the future where patients recovering from traumatic brain injuries, stroke, or progressive neurodegeneration are no longer told, "There's nothing we can do." Instead, they will have access to therapies that rebuild lost connections, restore cognitive function, and reclaim their independence. Quiver Bioscience has developed the technology and expertise to pursue these goals, but translating them into patient-ready treatments requires collaboration, funding, and vision. We invite you to join us in shaping this future. With your support, we can accelerate the development of life-changing therapies and bring renewed hope to millions affected with brain injuries. Thank you for your time, and I look forward to discussing how we can work together to make this vision a reality.